Science College, Brahmi, Kadameshwar, District Nagpur, Maharashtra. A chief guest of today's seminar, respected Mr. Satish Dalal sir, Head and Associate Professor, Department of Zoology of Nutan Adarsh College, Umar. IQC Coordinator, Dr. Sanju Kondekar sir of Nutan Adarsh College, Umar. Convener, Ms. Madhuri Varma Madam, Assistant Professor, Department of Zoology of Nutan Adarsh College, Umar. Organizing Secretary, Dr. Chandrahasya Nandanwar sir of Department of Physics, Assistant Professor of Nutan Adarsh College, Umre. Organizing Committee, Dr. Shailendra Khambadkar sir, Ms. Pranali Hinge Madam, Mr. Nilesh Chaple sir, Head and Assistant Professor, Department of Physics in the Nutan Adarsh College, Umre, and Dipti Ambildukar Madam, Assistant Professor, Department of Zoology. I welcome all the dignitaries of these auspicious occasions of one-day national seminar on innovative science in biophysics. I quote a line of Theodore Levity: creativity is thinking up new things, innovation is doing new things. To elaborate today's theme of seminar, I would like to invite Ms. Madhuri Varma Madam, Assistant Professor, Department of Zoology of Nutan Adarsh College, Umre, to deliver her introductory speech on innovative science in biophysics.
Thank you, Hindi ma'am. Respected chairperson of today's seminar, Dr. Chaudhary ma'am, Principal, Nutan Adarsh College, Umred. Respected resource persons, Dr. Ritesh Kohre sir and Dr. Asidhara Dharvikar ma'am. Chief guest, Mr. Satish Dalal sir. IQAC coordinator, Dr. Sanju Kondekar sir. Organizing secretary, Dr. Nandanwar sir. Members of organizing committee, all my respected colleagues and all the participants, I welcome you all. It gives me immense pleasure to welcome you all to this one-day national seminar on the theme Innovative Science, Accessible Tools in Biophysics. As a convener of today's program, I feel both honored and delighted to see such a vibrant gathering of scholars, educators, and young minds who share a common passion for exploring science at the interface of zoology and physics. Biophysics combines biology and physics to help us understand life at the molecular and cellular level. But often the tools we use are seen as costly or complicated. Today's seminar is about breaking that barrier, showing how innovative and accessible tools can empower students, researchers, and teachers alike. Even students and young researchers can participate meaningfully. We are living in a time when science is not locked in a big laboratories. It is becoming open, affordable, and reachable to all. These accessible tools have the power to ignite curiosity, train future scientists, and even contribute to translational outcomes in healthcare, agriculture, environmental sciences, and many more. I extend my heartfelt gratitude to our invited speakers who will be sharing their expertise and insights. I thanks all the participants joined here. I also I also thankful to organizing committee for making this event possible. I encourage each of you to actively participate in the sessions and engage in meaningful discussions. Let us use this platform not only to exchange knowledge, but also to inspire collaborations that can shape the future of accessible innovations in bioscience. I'm sure that the discussions today will spark new ideas that take biophysics forward in an inclusive way. With these words, I once again welcome you all and declare the seminar open. May it be a fruitful and enlightening experience for everyone. Thank you so much.
Thank you, Madam, to sparkling the light on biophysics with your marvelous words. Innovation is a vehicle to change everything. It can create a multiple copies of one piece. Before we proceed, let me take a moment to appreciate the guiding force and inspiration behind this event, our principal, Honorable Dr. Archana Chaudhary Madam, for organizing such a glorious event on such a vital theme of seminar. With this, I am pleased to invite Honorable Dr. Archana Chaudhary Madam, of Principal of Nutan Adarsh College, Umre, to deliver her inspiring words.
Thank you, Inge Madam. Am I audible?
Yes, ma'am.
Okay. So good morning all. Resource person for today's seminar on innovative science, accessible tools in biophysics, Dr. Ritesh Kore and Dr. Ashidhara Dharvikar Madam. Our head of the zoology department and senior faculty member, Dalal sir. Our IQC coordinator, Dr. Sanjeev Kondekar sir. Convener of this workshop, Ms. Madhuri Varbe, and Dr. Chandras Nandanwar sir, and all the organizing team and all the participants from different colleges of the region. I welcome you all to this seminar. As a principal, I take this opportunity to welcome you all to this seminar. And I really would like to congratulate the organizers for selecting such a wonderful topic on accessible tools in biophysics. Like we know about the sciences, physics, chemistry, maths, as a different subjects, biology, like botany, zoology. But nowadays it is a world of interdisciplinary science. And if you look at from the research point of view, for research, you need to collaborate with different subjects to reach to a particular conclusion. So this biophysics is an interdisciplinary science where it applies the principle and methods of physics to understand the biological system, how biological system works. And there are so many different perspective to look at this point of biophysics. Like biophysics, there are various aspects like structural biophysics, cellular biophysics, molecular biophysics, neurobiophysics. So many different branches are there, sub branches are there. So when you study this, like nowadays, you know that so many lifestyle diseases are there, and cancer is one of the disease which is growing at a very fast rate. So this biophysics helps us to apply the tools of physics to understand how the healthier cells are converted into the cancerous cells, what transportation mechanism is there, how cellular movement is there. And for this, they tag these cells with fluorescence molecules, and then they study accordingly. So to study this, there are various tools are required for this. So different tools are required, like we go for checking to any hospital for any medical purpose. We know that so many different tests the doctor advises us. So like the nowadays what we know about the MRI. It is magnetic resonance imaging. It is nothing but the application of NMR, that is nuclear magnetic spectroscopy. And this works on the principles of physics. And chemistry is also involved there because it works on the presence of water molecule in our body. And so it's an interface basically. And the principle of physics helps us to know what different changes, what are the changes which are happening in our body. So these tools are very, very important. And like Madam rightly said, the tools are very expensive. So all these tests are also very expensive. But what are the accessible tools? So this topic is very, very important. Like we know, X-ray crystallography. Whenever we visit doctor for some fracture or for some problem, the doctor advises to do the X-ray. So what is this? This X-ray is application of physics which understands our system, our biology, that what happened inside our body. And nowadays it's a world of this computation. So this microscopy is also very important. Spectroscopy is also very important. Then electrophysiology is also very important. And very important is mathematical modeling and simulations. Like the doctor studies various different patients and they do the mathematical modeling and simulation, and then they come to conclusion that what happens if the result is like this, then what happens inside the body. And so basically now it is a world of AI, there's a bioinformatics and big data analysis also plays a very, very important role in this. So now everybody should understand, basically the student should understand, and like NEP also says, it gives more stress on the interdisciplinary studies. So because today's seminar is about, there's a biophysics, that is combination of biology and physics, how they work together. Like this, all other subjects also, if you combine all the subjects together, then you get a wonderful results. So we look forward to our resource person, Dr. Ritesh Kore and Dr. Ashidhara Dharvikar, their lectures, which will definitely add value to the knowledge of the participants. And I hope that the seminar, the deliberations in this seminar will definitely prove fruitful to all the participants, and some of the participants may get inspired to do the research in this field. So once again, I congratulate all the organizers and I wish all the best for this seminar. Thank you.
Thank you, madam, for your kind and inspiring words. A ray of light is a ray of hope. I would like to invite Dr. Chandrahasya Nandan Varsar, Organizing Secretary, Assistant Professor of Department of Physics, to introduce a resource person of today's one-day national seminar, Dr. Ritesh Kore, sir.
Thank you, madam. I am delighted to introduce Dr. Ritesh Kore, who is presently working as an assistant professor and head Department of Physics, Sant Gadge Maharaj College, Hingna, Nagpur. He completed his MSc in Physics in 2008 and PhD in Luminescence in 2014 for RTMI University, Nagpur, and also holds an MA in Rural Development from IGNOU. With 11 years teaching experience, he has published more than 48 research papers, national and international journals, and presented over 12 research papers in conferences. He has authored 4 international books published by Opus and Web of Science, and written 2 textbooks for a graduate student, Fundamental of Nuclear Physics and Phosphorus Handbooks, which are selected as a reference books for MSc courses at Godman University. He has also published an Indian patents. He is a life member of several academic bodies, editorial boards member of the American Journals of Optics and Photonics, and reviewer for reputed journals like Elsewhere, Web of Science, Wiley, and RSC. His research contributions are even reflected in NASA ads. He visited abroad countries like Greece, China, and Hong Kong for a research and educational purpose. Currently, he is a master trainer at the Maharashtra State Faculty Development Academy and has conducted several workshops on science in daily life for school and college students. Now I request to such a versatile personality to guide us with his inspiring words.
Thank you very much, sir, for this wonderful introduction. Uh, at the outset, I'm very much thankful for giving me this opportunity to present my experiences throughout the daily life while teaching. I teach in the classrooms and labs. So I'm fortunate to present my experiences in one day national seminar on innovative science and accessible tools in biophysics. In the morning, I was talking to Dr. Chandrasi sir. So that is it a lecture or is it something interaction between the participants and students? So I'm interested to interact with the students and faculty members. I'm not today here to giving some sophisticated lecture in one day national seminar. I'm here to share my experiences with you all. So the management committee of this Sanstha, Principal Dr. Archana Chaudhary Madam, to whom I am interacting after a long while. I was there at Bhagwati Chaturvedi Engineering College and I learned a lot from Madam. So thank you so much, Madam. All the faculty members from various institutions amongst India and my dear students. let us start with some interesting facts in the physics and biology. So this seminar is jointly organized for biophysics. The name is given biophysics. So as Dr. Archana Chaudhary madam talked that no subject is specified in this era. It's a quantum age. Every subject is interrelated to one another. So we cannot bifurcate that it's a part of biology, it's a part of physics, it's a part of chemistry, it's a part of mathematics. Everything is interlinked with one another. And today we will proceed. So myself, Dr. Ritesh Kole, Assistant Professor and Head Department of Physics, Sant Gadge Maharaj Mahavidyalaya Hingna, which is located in Nagpur district, Hingna. So this is my institute where I used to teach and learn a lot. So basically, I'm inspired and accelerated by Maharashtra State Faculty Development Academy, ISER Pune, who conducts various workshops and seminars and faculty development programs for faculty members to inculcate their knowledge and to innovate something for students. And I'm very much inspired by Padma Shri Arvind Gupta sir and his foundation for continuously enhancing the concepts, the thoughts, the ideas for how to teach the students from daily life things. I'm also inspired by this mad guy for physics. He is always ready to die for physics, for the love of physics, Walter Levin. As you can see in the picture, he was performing some Newton's second law of motion demonstrations in front of his student, and the ball you are seeing in his hand is of iron ball, which is of 25-30 kg. When it comes to him, there was a risk. He can die also. It can beat to his head also. But he is also interested. He for the love of physics. Paul Abbott, who is a very good teacher as far as I concern, his idea, his thoughts, his way of teaching is very much good for students. And Mr. Ravindra Godbole sir, he is also an Indian teacher, especially in the field of astrophysics. So, my dear friends, physics is hopefully simple, but physicists are not. Edward Teller says, कि physics समझने के लिए easy है, but physicists को आप समझ नहीं सकते. They are always mad about their subject. They are always mad about their concepts. So physics and the most fascinating entity on this beautiful planet, on this living planet is our Mother Earth, what we call Mother Earth. There are lots of interesting events that takes place amongst us, around us. We never think about that events, that why sky is blue, why sky is orange, why it happens, why in the summer there is a huge intensity, and why in winter seasons, why there is a down in temperature. So these events are always happening around us. So how we can take them into our classrooms, take them into our labs in front of a student? It's very easy thing. At some point, your curiosity would have pushed you to ask some questions. Why, when, where, how, what's going on? How does that happen? ये ऐसे कैसे हो रहा है? इसके पीछे क्या reason हो सकता है? So always we used to ask the question to ourselves, but we never try to find the answers which are around us. So well, leaving miracles apart, the answers to all these questions is physics. And in fact, physics govern our everyday lives in day-to-day life. Let's have some examples in our daily life. So, my dear students, we always talk about light, that what is light? So, in very sophisticated definition is that light is electromagnetic waves that are sensible to human eyes, which travels with the speed of light. That is 3 into 10 raised to the power 8 meter per second, and we get the marks in the exams. But exactly, are you comfortable with English, or I should continue with English, Marathi, and Hindi? I think that will be more comfortable for students. Sir.
Yeah, sir.
Okay, sir.
Put the
Okay, sir.
time.
No problem.
सो हम हमेशा लाइट के बारे में बात करते हैं, क्या किसी ने लाइट देखा है स्टूडेंट्स? मैं आपको देख रहा हूं स्टूडेंट्स, यस एनी स्टूडेंट्स इन दिस सेमिनार? Am I visible to you? Me
Yes, sir,
display
you are
again, na?
visible.
Yes, sir. Yes, sir.
Okay, so what will be the
Yes, sir,
reason?
yes,
Please
sir.
keep your microphones on. Please keep your audio on. So मैं क्यों दिख रहा हूँ students आपको?
Due to reflection of light.
Right. So if I stop the light, all the lights, you cannot see me. So how could you define the light? Light को हम कैसे define करेंगे? Can we say light makes the things visible? Yes, we can say that light makes the things visible, but we cannot see the light. Light कहाँ है हमें दिखता नहीं है, वो है लेकिन दिखता नहीं है. तो how to see the light? How to define the light? Light makes the things visible, but light itself is invisible. So can you see this laser? मेरे हाथ में एक लेजर है, मैंने इसको ऑन भी किया है स्टूडेंट्स. सर प्लीज लाइट सर, आपको दिख रहा है कुछ? ओके, कैन यू सी? कैन यू सी नाउ? It's blinking.
Yes, sir. Yes, sir.
Okay. So मेरे हाथ से मैं स्क्रीन पे फोकस कर रहा हूँ इसको, लेकिन वो दिख ही नहीं रहा, बीच में कहीं दिख नहीं रहा वो. See, right? So how to see this light? Now you can see the light. Can you see, student? Something is yes or no? It's a laser. So we require some medium. If you want to see the light, we require some medium. हमें लाइट को देखने के लिए कुछ मीडियम चाहिए स्टूडेंट. Without medium you cannot see the light, and for that we require some dense medium. Let us have an example that why sky looks. So, light is basically electromagnetic waves that are sensible to human eyes, which travels with the speed 3 into 10 to the power 8 meter per second, जो होता है 3 लाख किलोमीटर पर सेकेंड. ओके, सो इसी लाइट से हमारे डे टू डे लाइफ में इतनी सारी फिनोमिना रिलेटेड है. जब हम सुबह उठते हैं तो हम देखते हैं कि द स्काई इज क्वाइट रेडिश, देन इट बिकम्स ऑरेंजिश, देन इट बिकम्स क्वाइट येलोइश, और 12 बजे आप जब देखोगे दोपहर में इट लुक्स लाइक व्हाइट. So there are so many reasons given by scientists, that is it's because of scattering of light, it's because of shifting of wavelength. So exactly how this waves scatter from the medium. So let us consider this is our sun on the left hand that you are seeing, and this is our mother earth. So from the sun there are different kinds of radiations continuously emitted because of the nuclear fusion and fission processes takes place on the sun. So gamma rays, x-rays, uv, then violet, indigo, blue, green, yellow, orange, red, and then infrared, microwaves, and radio waves, and so many cosmic radiations emitted from the sun. But from that only we can see this part of light that you call the Vibgyor spectrum, that you call the light, that you define the light that which are sensible to human eyes. हमें ये सन से जो पूरा का पूरा स्पेक्ट्रम आता है लाइट का, उसमें से सिर्फ ये 7 कलर के रेडिएशंस which is a combination in combination that becomes white light, यही हम देख पाते हैं बिकॉज हमारी आंखों का रेटिना उस तरह से बनाया गया है कि वो उस उतनी ही वेवलेंथ उसमें से सेंस कर सकते हैं. So why it looks, it is very simple phenomena. There is one very well known equation that E is equal to sc by lambda. Lambda is the wavelength. So forget wavelength, just keep in mind it's a length of wave. So जो waves ज्यादा travel करेगी वो हमें red दिखेगी, जो waves कम travel करेगी वो हमें bluish or ultraviolet दिखेगी. So the situation is somewhat like this. As you can see, let us prepare the medium here. I'm going to pour the water inside this measuring cylinder. Let us consider this is our atmosphere. ये हमारा एटमॉस्फियर है जिससे आपका सनलाइट ट्रैवल होता है. अब इसमें ये ट्रांसपेरेंट मीडियम दिख रहा है स्टूडेंट्स आपको, राइट? इस ट्रांसपेरेंट मीडियम से जब आपका लाइट हम इसको थोड़ा सा डेंस बनाते हैं. इसमें मैं थोड़ा सा मिल्क पोर करने जा रहा हूं. See, now the medium is preparing. आपको दिख रहा है स्टूडेंट्स? Please respond so that I can understand.
Yes,
Hello.
sir. Yes.
Why
Yes, sir.
the color of sky looks blue? So पहले हमने इसमें पानी लिया, उसमें थोड़ा सा मैंने मिल्क ऐड किया, सो हमारा अभी ये मीडियम बन गया. अब हम इस पे आपका सूरज जो सोर्स ऑफ लाइट है आपका, दैट वी विल स्टार्ट हियर. आप देख पा रहे हो उसको? सो नो? जस्ट अ मिनट. Now you can see here there is quite whitish. और आप ऊपर इसके ऊपर जब देखोगे it looks yellowish, right? So when light travels a lot, the wavelength increases and the color of this white light also increases. Let us have another example. ऐसा क्यों होता है? Can you see the setup, my dear students? So let us consider,
Yes, sir.
this is your sun and this is your position. This is Mother Earth. Okay, and this is you on the earth. Now, ये सुबह के 6 बजे स्टूडेंट्स, या फिर 5 बोलो, या फिर 4 बोलो, early in the morning, the distance of the observer, you are the observer, it's greater. So you look this sun reddish दिखता है या फिर थोड़ा सा orangish दिखता है आपको. अभी सुबह के 7 बजे, 8, 9, 10 ये आप हो अर्थ पे, 11, 12 ये दोपहर के 12 बजे. Now you can see the distance of the observer. ये डिस्टेंस कम हो गया स्टूडेंट्स सन से, राइट? पहले यहां थे, ये डिस्टेंस ज्यादा था, लाइट को ज्यादा ट्रैवल होके आना पड़ रहा था आपसे. हायर वेवलेंस मींस लोअर एनर्जी, हायर वेवलेंस मींस रेडिश कलर दिखता है. जैसे जैसे हम पास में आएंगे, सुबह के 12 बज चुके, आपको स्काई का कलर व्हाइट दिखता है या थोड़ा सा ब्लूइश दिखता है, बिकॉज द डिस्टेंस बिटवीन द ऑब्जर्वर एंड द सन डिक्रीजेस. अगेन इन द इवनिंग ये इस तरह से कुछ पोजीशन होती है आपकी. फिर से ये डिस्टेंस बढ़ गया स्टूडेंट यहाँ पे, isn't it? Again लाइट को यहाँ से ज्यादा ट्रेवल करके आना पड़ेगा यहाँ तक. So you will look the sky the orange color. So for sunset and sunrise, the color of sky is quite reddish or orange, and during the daytime, in the noon, it is quite whitish. So this is nothing but the shifting of wavelengths. ये दूसरा कुछ नहीं है, ये wavelengths का shifting है from in between the source and the observers. सुबह आप यहाँ होते हो, डिस्टेंस ज्यादा होता है. अर्ली 12 बजे आप यहाँ आते हो, डिस्टेंस कम होता है, वेवलेंथ डिक्रीज होती है. लेंथ डिक्रीज होना मतलब ही वेवलेंथ डिक्रीज होना, एंड इंक्रीज इन एनर्जी. हायर एनर्जी मींस ब्लूइश कलर. सो दिस इज वन ऑफ द मोस्ट 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 फैसिनेटिंग फिनोमेना इन डेली लाइफ. हम रोज देखते हैं अर्ली इन द मॉर्निंग, in the evening, but we never think in that point of view, physics point of view, biology point of view. Now another thing, that is waves. Sound waves है, light waves है, mechanical waves है. There are different kinds of waves. So हम इस wave को कैसे define करें? So let me confess here, all the participants and dignitaries, I designed this lecture especially for students point of view, so that how we can interpret our ideas in classrooms. सो स्टूडेंट्स, हम वेव को कैसे डिफाइन करेंगे? क्या है सिंपल डेफिनेशन वेव्स की? यस, एनी स्टूडेंट? I think nobody. So waves are the oscillatory disturbances in the motion of the particle. ये ऐसी इसकी बड़ी sophisticated physics की definition. कैसे oscillate होते हैं particles? कैसे disturb होते हैं वो? So see, the matter is made up of atoms and molecules which are in solids. They are tightly bonded with each other. And once you apply any force, for example, I am having this kinds of chain of particles. You can see these particles are attached to each other. If I disturb them, it will form a wave. If I disturb them, it will form a wave like this. Just a minute, sir. I will go. See. Can you see? Can you see wave? So you can see also the particles are vibrating also. Particle vibrates about their mean position. This is very simple phenomena in our life. So there are different kinds of waves depending upon the density of waves. So अगर wave को हमें देखना है, identify करना है, so this is the simple wave model that we prepare in our lab. आपको वेव प्रोपोगेट कैसे होती है स्टूडेंट्स मीडियम से आपको अगर देखना है, तो यू कैन सी लाइक दिस. सो दीज आर सिंपल स्ट्रॉज जिसको हमने एक आपण ज्याला चिकटपट्टी म्हणतो, ब्लॅक टेप है, त्यांनी असे स्ट्रॉज कनेक्ट केले. ये प्लास्टिक मीडियम है, ये थोड़ा सा लाइटर मीडियम है, इसकी डेंसिटी कम है. इफ आई स्ट्राइक समवेयर यू कैन सी द वेव. श्रुती लिंगायत. ओके, मी इथे फोर्स देतोय स्टुडंट. बघा, तुम्हाला त्या ब्लॅक स्ट्रिप वरनं वेव्ह ट्रॅव्हल होताना दिसत असेल. प्रोपोगेट्स फ्रॉम द मीडियम. सो इस तरह से आपकी वेव्स प्रोपोगेट होती है मीडियम से. एक्चुअल वेव्स इस तरह की होती है स्टूडेंट. द पार्टिकल्स मूव्स लाइक दिस. कैन यू सी? एंड इट्स अ स्पाइरल. जो आप ब्लैक बोर्ड पे देखते हो साइन वेव, कॉस वेव, इस तरह का फिगर, वो इस तरह की वेव्स होती है आपकी, राइट? इसका पिक्चर जब हम ड्रॉ करते हैं तो वो 2 डायमेंशनल पिक्चर ड्रॉ करते हैं, लेकिन ये 3 डायमेंशनल वेव्स को अगर हम देखेंगे, कंसीडर करेंगे, तो एक्चुअल पार्टिकल आपके इस तरह से मूव होते हैं. कैसे? ये उसका क्रस्ट, ये उसका ट्रफ, ये उसका क्रस्ट ट्रफ. सो इट इज अ क्रस्ट एंड ट्रफ. लेकिन इसको हम ब्लैक बोर्ड पे जब ड्रॉ करेंगे तो इट लुक्स लाइक दिस wave. So waves are nothing but these are the oscillatory disturbance in the motion of the particle. और वो oscillatory disturbance continue करते जाते हैं particle, one particle to another and so on. So there are different kinds of waves as you can see on the screen. Ultrasound waves है, infrared waves है, microwaves है, radio waves है. So it's a whole electromagnetic spectrum, starts from gamma rays and ends with the radio waves. 98.3 रेड एफएम आपने सुना होगा. that works on the radio waves. उसकी वेवलेंथ है जो आप देखते हो 98.3. सो आधीच्या काळात ते यायचं आकाशवाणीवर, की आकाशवाणीचे नागपूर केंद्र आहे 100.6 मेगाहर्ट्ज वरनं आम्ही बातम्या प्रक्षेपित करत आहोत. सो दॅट 100.6 मीन्स 100.6 megahertz was the frequency of that radio wave which propagates through the medium. So it's a kind of electromagnetic waves. All the waves are electromagnetic waves एक्सेप्ट मैकेनिकल वेव जो है आपकी, जो आप फोर्स देके जो मंदिर में आप घंटी बजाते हो साउंड आता है, या फ्लूइड से जो आता है, दीज आर मैकेनिकल वेव्स दैट कन्वर्ट्स इनटू द साउंड वेव्स. नाउ let us align with the syllabus of Bachelor of Science students for first year, second year, and final year. There are so many concepts in the physics and biology. We learn it exam point of view. We write it down in the exams. We get the marks. We get the degree. But till we don't understand what is the relation between these concepts, what are the importance, and what are the day-to-day applications of this concept. Kinetic energy and potential energy हमने सुना है students. Kinetic energy का general formula होता है half of mv square, that is the mass of particle, and the square of its velocity. And the potential energy, that is also called the gravitational potential. You can see on the screen, it is given by mgh. So this m is mass of the object, g is the acceleration due to gravity, and h is the height of the object from the altitude or sea level. सी लेवल से उस ऑब्जेक्ट की हाइट कितनी है. तो ये दोनों में बड़ा ही पैरा रिलेशन है स्टूडेंट्स. दे आर इनवर्सली प्रोपोर्शनल टू ईच अदर. जब आपकी पोटेंशियल एनर्जी हाई होगी तो काइनेटिक एनर्जी लो होगी. जब काइनेटिक एनर्जी हाई होगी तो पोटेंशियल एनर्जी लो होगी. सिंपल एग्जांपल है उसका वाटर टैंक. अगर आपकी वाटर टैंक थर्ड फ्लोर पे रखी है तो पानी नीचे फोर्स से आएगा. अगर आपकी वाटर टैंक फर्स्ट फ्लोर पे रखी है तो पानी का फोर्स कम रहेगा. जितना आप हाइट बढ़ाओगे उतना पोटेंशियल एनर्जी बढ़ेगी. पोटेंशियल एनर्जी बढ़ने से जब आप काइनेटिक एनर्जी स्टार्ट करते हो, आपकी पानी की टैप जब स्टार्ट करते हो तो काइनेटिक एनर्जी बढ़ती जाती है. वाटर टैंक का पानी कम होते जाता है, मतलब पोटेंशियल एनर्जी डिक्रीज होना. सो दिस इज वन ऑफ द बेस्ट एग्जांपल फॉर रिलेशन बिटवीन काइनेटिक एनर्जी एंड पोटेंशियल एनर्जी. सो उसका कर्व जो आप यहाँ पे देख रहे हो ऊपर, there is an inverse relation. Let's have a demonstration to visibly see the relation between the kinetic energy and potential energy. So my dear students and participants, ये दो bottles है जो एक ही दुकान से खरीदी है. सेम इसका वॉल्यूम आएगा, इसमें सेम साइज है, सेम प्राइस है, सेम कलर है. दोनों में एक सरी का पानी मैंने भर के रखा है यहाँ पे, सेम यहां तक, इसकी नेक तक, राइट. और इसकी कैप्स को अभी हम लगा देते हैं स्टूडेंट्स. ये बॉटल्स की कैप्स को लगा देते हैं. दोनों में सेम अमाउंट ऑफ वाटर हमने भर के रखा है. दोनों को ये कुछ ट्यूब्स लगाई है. डियर स्टूडेंट्स, प्लीज on your audios, I'm going to ask the questions. अगर मैं इसको उल्टा करूं दोनों बॉटल्स को, ये दोनों जो ट्यूब्स आपको दिख रही है इससे पानी बाहर आएगा, तो कौन सी बॉटल पहले खाली होगी? देखो इसमें डिफरेंस क्या है स्टूडेंट्स. ये दोनों सेम लेंथ की है. इसकी लेंथ थोड़ी ज्यादा है, इसकी लेंथ थोड़ी कम है. How to understand the relation between the kinetic energy and potential energy? आपको अभी चेक बताना है कि ये दो बॉटल में से पहली बॉटल सबसे पहले कौन सी खाली होगी या कौन सी ज्यादा खाली होगी at the same time. Yes, any answers? Let's make it interactive, students. Actually, this lecture or this workshop is not doesn't suit online. It's a hands-on training for students. बट अभी हमारे पास कोई ऑप्शन नहीं था, इसलिए ये ऑनलाइन प्रेजेंट करना पड़ा है. अदरवाइज जब हम ये लाइव देखते हैं, सो वी फील दैट साइंस, वी फील दैट फिजिक्स, वी फील दैट बायोलॉजी. यस, एनी स्टूडेंट्स? यस, एनीबॉडी एल्स? दो में से कौन सी बॉटल सबसे पहले खाली होगी अगर हम इसको उल्टा कर देते?
सर स्मॉल पाइप वाली.
ये ओके, या फिर ये
सर स्मॉल पाइप.
ये खाली होगी पक्का?
हाँ.
सी स्टूडेंट्स, इसके दोनों ये जो पाइप्स है ये दोनों सेम लेंथ वाले हैं. इसकी लेंथ थोड़ी कम है, इसकी लेंथ थोड़ी ज्यादा है. स्क्रीन पे आपको काइनेटिक एनर्जी और पोटेंशियल एनर्जी के इक्वेशंस दिख रहे हैं. हमको वो जीना है, हमको वो फील करना है, हमको एक्सपीरियंस करना है इक्वेशंस। हमें सिर्फ एग्जाम्स में नहीं लिखना है स्टूडेंट्स दैट P is equal to mgh and K is equal to half of mv square. Let us see practically whether it works or not. Potential energy is inversely proportional to the kinetic energy. Let's start. Now I am going to pour this water. ये दोनों सेम बॉटल, सेम साइज, सेम वॉल्यूम, सेम वाटर, सिर्फ एक पाइप को हमने छोटा बड़ा किया है. So let us check. Shall I start?
Yes, sir.
Shall I start? कौन सी बोतल खाली होंगी? किसने आंसर दिया था? छोटे पाइप वाली ना,
हाँ Yes, सर sir. मैंने दिया था।
जो मेरे लेफ्ट हैंड में ये
हाँ Yes, sir. सर।
ये खाली होगी पक्का?
हाँ हाँ सर। यस सर।
बड़े पाइप वाली होगी जिसने भी आंसर दिया था. यू कैन सी. सो क्या रीजन है होगा स्टूडेंट्स इसका? ऐसा लगता है हमें कि छोटे पाइप वाली जल्दी खाली होंगी. देयर इज नो एनी साइंटिफिक बेस. हम ऐसे आंसर दे देते कि मुझे लगता है कि ऐसा, बट वी हैव साइंस, वी हैव फिजिक्स, वी हैव द थिंग्स टू प्रूव इट. साइंटिफिक टेम्परामेंट हमें आनी चाहिए एज वी आर द स्टूडेंट्स ऑफ साइंस. सो वो चीजें हमें इंप्लीमेंट करना आना चाहिए. सी mgh वाला जो इक्वेशन दिख रहा है ना आपको सेकंड, सो h है वो हाइट. अब जब हम इस बॉटल का सोचेंगे, just a minute. देखो स्टूडेंट, ये इस तरह का सिचुएशन था. जो ये बॉटल है इसकी हाइट कम है, राइट? ये सी लेवल से इसकी हाइट कम है और इसकी हाइट ज्यादा है. इसकी पोटेंशियल एनर्जी ज्यादा, दैट्स व्हाई इट्स काइनेटिक एनर्जी इज लेस. इसकी पोटेंशियल एनर्जी ज्यादा, इनवर्स प्रोपोर्शन होता है स्टूडेंट. जिसका एच ज्यादा, जिसकी हाइट सी लेवल से ज्यादा थी, सो इसकी पोटेंशियल एनर्जी ज्यादा, काइनेटिक एनर्जी कम. पानी फ्लो होने का जो स्पीड था वो कम था इस बॉटल का. जो आंसर दिया था आपने वो ऐसे दिया था कि सर ये इसमें से पानी जल्दी खाली हो जाएगा, लेकिन पानी एक्चुअली इसमें से खाली हुआ क्योंकि इसकी हाइट कम है सी लेवल से. तो इसकी पोटेंशियल एनर्जी कम, काइनेटिक एनर्जी ज्यादा थी, पानी ज्यादा स्पीड से इसमें से आया. सो दैट इज द रिलेशन बिटवीन काइनेटिक एनर्जी एंड पोटेंशियल एनर्जी. डे टू डे लाइफ एग्जांपल है हमारा पानी की वाटर टैंक. जितनी ज्यादा ऊपर रखोगे आप, उतना स्पीड से, फोर्स से आपका काइनेटिक एनर्जी फ्लो होगा. Now let us align with the syllabus, especially for the final year students. There is a X-ray tube which was invented by William Röntgen. An X-ray tube, it is the vacuum tube that converts the electrical input power into X-rays. वो voltage को rays में convert कर देता है X-rays में, which are not visible. हम X-rays को देख नहीं पाते क्योंकि that are not sensible to human eyes. उसकी energy बहुत ज्यादा होती है. They can penetrate through your skin up to the bones. X-ray tubes are used in the city scanner, airport, luggage scanners जो आप अभी मेट्रो में देखते हो. एक्सरे, क्रिस्टलोग्राफी, मटेरियल का स्ट्रक्चर्स और उसमें डिफेक्ट्स क्या है वो आइडेंटिफाई करने के लिए एक्सरे का यूज होता है. सो बेसिकली दिस एक्सरे ट्यूब इज कंसिस्ट ऑफ द वैक्यूम ट्यूब मेड अप ऑफ फिलामेंट एंड द वोल्टेज सोर्स एंड द वोल्टेज रेगुलेटर. हम एग्जाम्स में पढ़ते भी है, लिखते भी है, रट भी लेते हैं इसको, लेकिन हम इसको देख नहीं पाते. इट इज नॉट पॉसिबल टू ब्रिंग दैट एक्सरे ट्यूब इनसाइड आवर क्लासरूम्स एंड टू शो द स्टूडेंट्स. तो हमने इसमें एक इसी से मैच होने वाला, इसी के जैसे काम करने वाला एक एक्सरे ट्यूब बनाया है, जो एक्चुअल एक्सरे ट्यूब नहीं है. जैसे एक्सरे ट्यूब से एक्सरे बाहर आते हैं, हमारी ट्यूब से लाइट बाहर आएगा. एक्चुअली एक्सरे भी आपका इलेक्ट्रोमैग्नेटिक रेडिएशन ही है और आपका विजिबल लाइट भी इलेक्ट्रोमैग्नेटिक रेडिएशन ही है. उनमें फर्क इतना है स्टूडेंट, इनकी वेवलेंथ में डिफरेंस है, इनकी एनर्जीज में डिफरेंस है। विजिबल लाइट की वेवलेंथ 4000 टू 7000 एंगस्ट्रॉम है, और एक्सरे की वेवलेंथ 1 एंगस्ट्रॉम टू 2 एंगस्ट्रॉम के बीच में होती है। लेकिन अगर हम ऐसा मॉडल देखना चाहे कि एक्चुअली ये कैसा वर्क करता है, सो वी कैन प्रिपेयर दिस मॉडल बाय वेरी सिंपल टेक्निक। लेट अस चेक क्या होता है इसमें। फिगर दिख रही है सभी को? इसमें एक ये वैक्यूम ट्यूब होता है स्टूडेंट, ये आपका टारगेट मटेरियल है जो ऑरेंज कलर का दिख रहा है, और इसको एक वोल्टेज सोर्स प्रोवाइड किया है, ऊपर में आप देख रहे हो पॉजिटिव और नेगेटिव, राइट। और यहाँ पे ये फिलामेंट जब हीट होता है, टारगेट को हीट करता है, तो इलेक्ट्रॉन्स का इंटरेक्शन होता है यहाँ पे, और यहाँ से एक्सरेज बाहर आते हैं। सेम थिंग हैपेंस। ये आपने देखा होगा सभी ने, यस और नो? ये ऐसा कुछ देखा होगा ना सभी ने, होल्डर है और उसको एक रेगुलेटर, इलेक्ट्रिक बोर्ड आपने देखा होगा ना स्टूडेंट्स। ये रेगुलेटर बेसिकली हम फैन को लगाते हैं। दिस इज वोल्टेज डिवाइडर और पोटेंशियोमीटर कहते हैं इसको, आपकी लैंग्वेज में आप इसको रेगुलेटर कहते हो, इजंट इट? सो एक्सरे ट्यूब में क्या होता है, पोटेंशियल इज डायरेक्टली प्रोपोर्शनल टू द इंटेंसिटी ऑफ एक्सरेज कमिंग आउट ऑफ दैट ट्यूब। ये जो एक्सरेज है ना, इसकी इंटेंसिटी, आप जितना पोटेंशियल बढ़ाओगे उतना उनकी इंटेंसिटी बढ़ती जाती है। वेरी सिंपल फिनोमिना। विलियम रॉन्टजन ने जब इन्वेंट किया था, दैट वाज नॉट सिंपल। उन्होंने जब फर्स्ट टाइम इन्वेंट किया था तो वो इतना सिंपल नहीं था। फिलामेंट, पोटेंशियल डिवाइडर अरेंजमेंट, उसमें से आने वाले रेज कौन से, उनको डिटेक्ट करना। तो हम क्लासरूम्स में, लैब्स में अगर समझना है एक्सरे ट्यूब का मैकेनिज्म क्या होता है, सो दिस इज सिंपल होल्डर एंड दिस इज टंगस्टन लैम्प। इसके अंदर फिलामेंट होता है स्टूडेंट, आप देख पा रहे हो? इसके अंदर जो स्प्रिंग जैसी तार होती है ना, वो टंगस्टन फिलामेंट होता है, दैट वर्क्स एज अ कैथोड। और यहाँ पे लगा दिया हमने इसको एक रेगुलेटर। ये जो वायर है, ये हमारा पोटेंशियल अरेंजमेंट है जो आप फिगर में देख रहे हो इसमें, है ना? ये प्लस माइनस, तो ये हमने बोर्ड से दे दिया है प्लस माइनस। नाउ आई एम गोइंग टू ऑन इट। फिलामेंट दिख रहा है आपको स्टूडेंट्स? हेलो हेलो,
Yes, sir. Yes.
am I audible?
Yes, sir.
ये फिलामेंट ग्लो हुए वाला दिख रहा है आपको? और स्क्रीन पे
हाँ
एक्सरे
सर,
ट्यूब
दिख
भी
रहा
दिख
है।
रहा होगा, स्क्रीन पे
हाँ
एक्सरे
सर।
ट्यूब भी दिख रहा है। तो वो
Yes,
एक्सरे
sir.
ट्यूब से जो ग्रीन कलर के एक्सरेज आ रहे हैं, ये हमारा लाइट है इसके। अब जैसे ही मैं पोटेंशियल बढ़ाऊंगा तो ये लाइट इंटेंस होते जाता है। यहां से मैं अभी पोटेंशियल बढ़ा रहा हूं, राइट? सो ये जो बाहर आ रहे हैं, लेट्स कंसीडर दिस आर और एक्सरे। जितना ज्यादा पोटेंशियल आप दोगे उतने ज्यादा इंटेंस एक्सरे उसमें से बाहर आएंगे। अब मैं पोटेंशियल कम कर रहा हूं स्टूडेंट। So intensity of X-ray is directly proportional to the potential applied to that filament. Now it is off. X-ray emit होना बंद हो जाएगा। आपने सोर्स ऑफ कर दिया, एक्सरे emit होना बंद हो जाता है एक्सरे ट्यूब से। सोर्स ऑन कर दिया, एक्सरे emit होना। सो लेट अस कंसीडर ये जो लाइट आ रहा है, इट इज अ काइंड ऑफ एक्सरे। यही एक्सरे है, यही रॉन्टजन का इन्वेंशन था जिसके लिए उनको नोबेल प्राइज मिला स्टूडेंट्स। सो इज इट अ सिंपल कंसेप्ट टू अंडरस्टैंड हाउ एक्सरे ट्यूब वर्क्स? As you increase the potential of filament, the radiations coming out of the intensity of radiations coming out of the tube increases, and as you decrease the potential, it also decreases. So this is very simple phenomenon which relates your X-ray tube, that is in your syllabus of 6th semester. Then आपने ये भी सुना होगा अल्फा, बीटा, गामा रेडिएशंस, नाम तो सुने होंगे ना स्टूडेंट्स? Did you know that you can see the traces of this alpha beta? अगर आपको विजिबली देखना है अल्फा बीटा गामा रेडिएशंस, तो कैसे देखेंगे स्टूडेंट्स? Don't worry, even I also don't know how to see it, but Wilson cloud chamber, that is in your syllabus of 6th semester. Wilson cloud chamber is the device, that is the detector which detects the tracks of the particle. आपको स्क्रीन पे दिख रहा होगा राइट मोस्ट कॉर्नर में गामा रेडिएशन स्ट्रेट ट्रेवल करते होते, डू नॉट अफेक्ट विथ फील्ड्स और पोटेंशियल्स। वो कोई भी फील्ड से, इलेक्ट्रिक फील्ड या मैग्नेटिक फील्ड से डिफ्लेक्ट नहीं होते। और अल्फा पार्टिकल्स दे आर पॉजिटिवली चार्ज पार्टिकल्स, वो नेगेटिव की तरफ अट्रैक्ट होंगे। एंड बीटा पार्टिकल्स दे आर नेगेटिवली चार्ज रेज और पार्टिकल्स, दे विल अट्रैक्ट टुवर्ड्स दी पॉजिटिव। अब इनको हमें देखना है एक्चुअल में कि ये कैसे होते हैं। अल्फा गामा तो हम देख नहीं पाएंगे, बहुत ज्यादा एनर्जी होती है, दे कैन पेनिट्रेट थ्रू योर eyes and they can burn your eyes, they can burn your cells, body cells, कैंसर्स भी हो सकते हैं। दैट्स व्हाई गामा से कोई खेलता नहीं है। दे आर वेरी हाई एनर्जेटिक रेडिएशंस, फंडामेंटल रेडिएशन जहां से स्टार्ट होते हैं वो है गामा रेडिएशंस। अल्फा बीटा यू कैन सी, अल्फा बीटा यू कैन ट्रैक दी पाथ्स। Now, this was the chamber, Wilson cloud chamber, which was built by scientist Wilson to identify the tracks of the particle. इसमें क्या होता है स्टूडेंट, ये एक मेटल सिलेंडर होता है, राइट? ये मेटल सिलेंडर के अंदर पिस्टन होता है जो ऊपर नीचे होता है, ऐसा पिस्टन। और यहाँ पे जो रेडिएशंस अल्फा बीटा गामा आपको डिटेक्ट करना है उसका सोर्स होता है, और यहाँ से उसके अंदर लाइट इंसिडेंट की जाती है। और ये कैमरा होता है ऊपर में उसका फोटो लेने के लिए कि अल्फा पार्टिकल्स कैसे दिखते हैं, गामा पार्टिकल्स कैसे दिखते हैं, बीटा पार्टिकल्स। सो ये फिगर में आप देख पा रहे हो, अल्फा रेडिएशंस कुछ इस तरह के थिक ट्रेसेस उसके मिलते हैं कैमरा में जब हम फोटो लेंगे। बीटा पार्टिकल्स कुछ ऐसे डॉट 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 लाइक शेप में दिखते हैं, दे आर लो एनर्जेटिक रेडिएशन। और गामा यू कैन सी दे लुक स्ट्रेट। स्टूडेंट्स, कैन यू सी द पिक्चर ऑन द स्क्रीन? सो कुछ नहीं है, विल्सन क्लाउड चेम्बर में एक मेटालिक सिलेंडर है, उसके नीचे एक पिस्टन है जो ऊपर नीचे होता है। अब यहाँ पे आपका गैस लॉ भी लगता है, और यहाँ पे सैचुरेटेड वेपर्स वगैरह लिखा हुआ है साइंस की लैंग्वेज में। ये धुआं है या फिर इथिल अल्कोहल का वेपर है या फिर सिंपल गैस वेपर है। क्या होता है जब कोई भी लाइट मीडियम से ट्रेवल करता है तो वो दिखता है, हमने हमने सबसे पहले देखा है लाइट, how we can see the light. So let us consider this is your metallic cylinder. अब ये प्लास्टिक का दिख रहा है बट इसे कंसीडर करो मेटालिक सिलेंडर है आपका। इसके नीचे जो पिस्टन लिखा हुआ है फिगर में, ये पिस्टन है जो ऊपर नीचे होता है, राइट? इस तरह से। और ये है आपका रेडियो एक्टिव सोर्स एंटर करने के लिए होल यहाँ पे जो आप इसमें देख रहे हो, और यहां से हम लाइट इंसिडेंट करेंगे, यहां से रेडियो एक्टिव सोर्स हम इंसिडेंट करेंगे या लाइट इंसिडेंट करेंगे, और ऊपर से लेंगे फोटोग्राफ। यहाँ पे हम हमारा मोबाइल का कैमरा रख देंगे और लाइट इसमें से जब ट्रैवल होंगी तो इसके लिए मीडियम होना, स्टूडेंट्स लाइट को देखने के लिए आपको क्या होना, मीडियम होना। हाँ, अब मैं देखो स्टूडेंट, जब इसमें से मैं लाइट पास करूंगा हमें दिखता नहीं है, इसके अंदर ट्रैक नहीं दिख रहा है लाइट का। ये लेजर लाइट में जब इस पे इंसिडेंट करूंगा दिखता नहीं। जब हम मीडियम क्रिएट करेंगे, अब हम इसके अंदर मीडियम क्रिएट करके इसके अंदर मैं थोड़ा सा अगरबत्ती लगा के फॉग डेवलप कर रहा हूं इसके अंदर। ये इथिल अल्कोहल की वेपर जैसा ही काम करेगा, अगरबत्ती का धुआं आपका। वी विल क्रिएट द फॉग इनसाइड दिस metallic cylinder. Then we will put this piston, जिससे आपको ट्रैक दिख रहा है स्टूडेंट, अंदर लाइट का ट्रैक दिख रहा है? अगर नहीं दिख रहा है तो मैं आपको फोटोग्राफ्स दिखाता हूँ। अभी थोड़ा सा रिफ्लेक्शन आ रहा है इसलिए आप नहीं देख पा रहे हो इसके अंदर से। सो इसी तरह के ट्रैक्स विल्सन क्लाउड चेम्बर में डिटेक्ट किए गए थे। ये सिर्फ मैंने जहाँ पे लेजर लगाया था यहाँ पे रेडियो एक्टिव सोर्स था। सो द ट्रैक्स वर लाइक दिस। ये जो दिख रहा है ना स्टूडेंट स्ट्रेट लाइन में लेजर, ये एक्चुअल हमारा जो मॉडल था एक्चुअल पाथ विल्सन क्लाउड चेम्बर की तरह ट्रैक करने के लिए, ये वो फोटोग्राफ्स है जो अल्फा बीटा गामा रेडिएशंस के विल्सन ने लिए थे। ये वो फोटोग्राफ्स हम भी ले सकते हैं। विल्सन ने रेडियो एक्टिव सोर्स यूज किया था, हमने लेजर लाइट यूज कर लिया। विल्सन ने मेटालिक सिलेंडर यूज कर लिया था, हमने प्लास्टिक सिलेंडर यूज कर लिया। ओके, विल्सन ने कैमरा लगाया था, हमने मोबाइल लगा दिया। बट इसका जो वर्किंग है, जब हम इसका प्रेशर इंक्रीज करते हैं तो टेम्परेचर डिक्रीज होता है। टेम्परेचर डिक्रीज होना मतलब कंडेंसेशन होता है वहां पे, जो भी इथिल अल्कोहल की वेपर है, और वो लाइट रे जो भी है रेडिएशन, उस पे वो ड्रॉपलेट्स जमते जाते हैं और उसका फोटोग्राफ आप इजीली ले सकते हो। सो दिस इज सिंपल मैकेनिज्म ऑफ विल्सन एंड क्लाउड चेम्बर। नेक्स्ट आपके सिलेबस में ही है, दैट इज GM counter. This is the counter to detect the number of particles entering inside the chamber. तो इसका इस तरह का मैकेनिज्म है स्टूडेंट। ये देखो यहाँ पे एनोड दिख रहा है आपको, ये एक रॉड होता है एनोड, और ये काम करता है एनोड जैसा पॉजिटिव, और एक मेटालिक ट्यूब, और ये इट एक्ट्स एज अ कैथोड, और ये दोनों को यहाँ पे करंट मीटर कनेक्ट कर दिया, और इधर से होता है गामा रेडिएशंस या अल्फा रेडिएशंस या बीटा रेडिएशंस इसके अंदर एंटर किए जाते हैं। ये फिगर आपके सिलेबस में है, बुक में है, बट ये मॉडल एक्चुअल कुछ इस तरह का है। सी, ये आपका ट्यूब, ये मेटल सिलेंडर, ये मेटल सिलेंडर, ये आपका एनोड, इसके अंदर जो रॉड दिख रहा है आपको, राइट, इस एनोड को ये कनेक्ट किया है, जो हमें भी इस मीटर को कनेक्ट करेंगे। इस ये इलेक्ट्रिक मीटर है, राइट, और ये मेटालिक सिलेंडर का एक कॉर्ड जो नेगेटिव काम करेगा, और यहां से इसके अंदर रेडिएशंस एंटर करेंगे स्टूडेंट, इस तरह से। बात समझ में आ रही है? तो जैसे ही रेडिएशन एंटर करेंगे अल्फा बीटा गामा, तो अंदर आयनाइजेशन होगा, सारे नेगेटिव चार्जेस इस रॉड के ऊपर कलेक्ट हो जाएंगे, सारे पॉजिटिव चार्जेस मेटल सिलेंडर के पास आ जाएंगे, और वहां पे पोटेंशियल डेवलप होगा। जो पोटेंशियल डेवलप होगा वो हमें इस मीटर में दिखेगा, दैट विल गेट कन्वर्ट इनटू द इलेक्ट्रिक करंट। सो जितना ज्यादा स्ट्रॉन्ग रेडिएशन सोर्स होगा आपका, उतना ज्यादा यहाँ पे करंट मिलेगा, और वो पल्सेस के फॉर्म में मिलेगा करंट। सो हाउ मेनी नंबर ऑफ पार्टिकल्स एंटरिंग, that will be directly proportional to the current in the current meter. So इस तरह का ये सिंपल मॉडल है गाइगर मूलर काउंटर, that you learn in sixth semester। जो 6th सेमेस्टर में स्टूडेंट्स होंगे उनको वो सिलेबस में है। सो इन दिस वे वी कैन प्रिपेयर आवर ओन मॉडल्स। अब आपने लिसाजस फिगर्स भी सुना होगा स्टूडेंट, ये सीआरओ पे हम हमेशा इसको देखते हैं, क्या हम एक्चुअल में इसको नहीं देख पाते? हमारे पास सीआरओ नहीं है क्लासरूम्स में, फॉर एग्जांपल हमारे पास लैब्स में सीआरओ नहीं है, तो हमें वैसे विजिबली हम फिजिकली नहीं देख पाते। हमने देखा है एक्चुअल वेव्स किस तरह की होती है स्टूडेंट, इस तरह की होती है वेव, जब ट्रैवल होती है मीडियम में तो इस तरह से ट्रैवल होती है। यस और नो? जो हम ब्लैक बोर्ड पे और बुक्स में फिगर्स देखते हैं, या फिर इंटरनेट पे आपने जो भी फिगर देखी होगी, वो इस तरह की नहीं है। वो वेव ऐसी होती है, ऐसी ट्रैवल करती है, 3 डायमेंशनल वेव अगर स्पाइरल वेव्स होती है। तो लिसाजियस फिगर्स 2 वेव्स के कॉम्बिनेशन से फॉर्म होती है, जो आप इस पे देख रहे हो स्क्रीन पे, 8 लाइक फिगर्स। तो ये जब परपेंडिकुलरली ट्रैवल होती है दोनों वेव्स एक में को, सो कैन यू सी द 8 लाइक फिगर्स? एक वेव इस डायरेक्शन में जा रही है पॉजिटिव एक्स डायरेक्शन में, और एक वेव पॉजिटिव वाई डायरेक्शन में ट्रेवल हो रही है। दिस इज नथिंग बट द इलेक्ट्रोमैग्नेटिक वेव का जो प्रोपोगेशन होता है स्टूडेंट मीडियम में, वो कुछ इस तरह से होता है। अगर आप, आई डोंट नो एंगल ठीक है कि नहीं, लेकिन 8 लाइक फिगर्स इसमें जो दिखती है वही हम सीआरओ में दिखते हैं। सो मैकेनिकली भी हम इस तरह से देख सकते हैं वेव्स को, कि वेव्स कैसे ट्रेवल होती है, तो ऐसे होती है स्पाइरली, राइट, पार्टिकल्स ऐसे ट्रेवल होते हैं। सबसे पहले एकदम शुरू में हमने देखा कि वेव्स क्या है, ऑसिलेटरी डिस्टरबेंस इन द मोशन ऑफ द पार्टिकल। तो हम लिसाजस फिगर्स भी फिजिकली देख सकते हैं विदाउट सीआरओ। सो इस तरह से आपने देखा होगा, ये जो फिगर दिख रही ना यहाँ पे, ये 8 लाइक फिगर, it is Lissajous figure. That is the combination of two waves. So ideal gas law. This is my, I think last. आइडियल गैस लॉ आपने सुना होगा स्टूडेंट, पीवी इज इक्वल टू एनआरटी। इतने सारे एप्लीकेशन है हमारे आइडियल गैस लॉ के डे टू डे लाइफ में। आपका डिओड्रेंट, आपका सैनिटाइजर, आपका हैंड वॉश की बोतल, आपका प्रेशर कुकर, आपके घर का सिलेंडर, आपकी सिरिंज इंजेक्शन लेने वाली, ऑल दिस थिंग्स वर्क्स ऑन very simple law. PV is equal to nRT, in which volume is inversely proportional to pressure at constant temperature. अगर आप temperature constant रखते हो, so volume is inversely proportional to temperature, n is number of gas molecules and R is your Rydberg constant. So इसको हम किस तरह से देख सकते हैं? हमारी डिओड्रेंट की जो आपकी कैन होती है, so that works on the principle of ideal gas law. That is day to day life example. So ये एक सिंपल मॉडल है स्टूडेंट्स। अभी ये एटमॉस्फेरिक प्रेशर पे है। Those who are attending this workshop, ये एटमॉस्फेरिक प्रेशर पे है। इसके अंदर मैंने एक बलून डाला है स्टूडेंट यहाँ से, राइट, और यहाँ पे एक ओपनिंग है। नाउ दिस इज ओपन एंडेड सिस्टम। इसका ये भी एंड ओपन है, इसका ये भी एंड ओपन है। अगर हम इसको क्लोज एंड कर देंगे और मैं प्रेशर कम करूंगा तो वॉल्यूम क्या होना चाहिए? इंक्रीज होना चाहिए। यस माय डियर स्टूडेंट्स, अकॉर्डिंग टू आइडियल गैस लॉ, इफ वी डिक्रीज द प्रेशर, वॉल्यूम इंक्रीज होना चाहिए। देखते हैं फिजिकली ऐसा होता है क्या। सो ये बॉटल अभी एटमॉस्फेरिक प्रेशर पे जो आप देख रहे हो, इसका ये ओपनिंग है जिससे हम प्रेशर सक करेंगे। यहां से मैं इसको एयर निकाल लूंगा तो प्रेशर कम होगा, बलून का वॉल्यूम इंक्रीज होता है क्या, हम चेक करते हैं। लेट अस सी। हुआ कि नहीं? हेलो स्टूडेंट्स, आर यू लिसनिंग? आर यू सीइंग? इज इट अ वन वे कम्युनिकेशन? आई डोंट लाइक दिस। Okay, Airpude sir is there. Okay, this is open-ended system. जब हम इसका प्रेशर कम कर लेंगे तो बलून का वॉल्यूम इंक्रीज हो गया। एक्चुअल बलून में हमने हवा नहीं डाली है, हमने यहाँ से हवा निकाली है। तो एटमॉस्फेरिक प्रेशर, कांस्टेंट एटमॉस्फेरिक प्रेशर पे आने के लिए उसने अपना वॉल्यूम मेंटेन कर लिया। सी, सो दिस इज आइडियल गैस लॉ। It satisfies when you decrease the pressure, when you suck the air, the volume of balloon increases. So pressure is inversely proportional to the volume. This is Joule-Thomson effect. Students, please turn on your cameras and audio. Students especially. Students, no student is there in this seminar. I think student just log in and they went away. Okay, participants can also. Okay, this is Uma madam. Good afternoon.
Good afternoon, sir. Very nice
so this is
presentation
very,
going on.
thank you, madam. This is very simple phenomenon, Joule-Thomson effect. When you compress the air, the temperature decreases. When you expand the air, the temperature increases. Let's do like this. You will feel that the air is hot, and just do like this. Compress the air. You will feel the cold air. So this is expansion and compression of air. This we can do in day-to-day life by using this concept. And in our physics and science, the name is again Joule-Thomson effect. We write it down in the exams, we learn it, we mug it, and we get the marks also. But till end, we don't know what was Joule-Thomson effect. So this is Joule-Thomson effect, dear students. So what are the applications of this Joule-Thomson effect? We can prepare the homemade AC by using this Joule-Thomson effect. The situation is somewhat like this. ओके, ये बॉटल को कट किया है, ये बिगर डायमीटर कहेंगे इसको, या फिर ये स्क्वायर है तो एरिया बोलेंगे, एंड दिस इज स्मॉलर। व्हेन यू पास द एयर फ्रॉम दिस एंड, when you pass the air from this end, the air will be hot, and when you compress at this end, the air will be cold. Okay, सो ये बॉटल्स को अगर ये एक्सपेरिमेंट्स हमने करके देखा है, इट वर्क्स। 2 से 3 डिग्री का डिफरेंस आता है एक्सटर्नल टेम्परेचर और इंटरनल टेम्परेचर में। जब समर सीजन में ऐसी बॉटल्स हमने विंडोज को लगा दी, बहुत सारी बॉटल्स, यहाँ से गर्म हवा आएगी, ये एंड ओपन है, आप देख रहे हो इसमें जब एयर एक्सपांड होती है तो गर्म होती है, और जब एयर कॉम्प्रेस होती है तो ठंडी होती है। तो ये इस तरह के हमने अगर बॉटल्स कट करके विंडोज में लगा दी, ये साइड से गर्म हवा आएगी और यहाँ पे कॉम्प्रेस होके अंदर आएगी तो कोल्ड होके आएगी। सो दिस इज द एक्चुअल एप्लीकेशन ऑफ जूल थॉमसन इफेक्ट इन डे टू डे लाइफ। लेट्स मूव फॉरवर्ड। So this is Joule Thomson effect in our kitchen also. Liquid petroleum gas, LPG कहते हैं उसको हम। बहुत से स्टूडेंट्स को मैं जब पूछता हूँ कि सिलेंडर में क्या भरा होता है, तो वो बोलते हैं कि गैस भरा होता है। एक्चुअल सिलेंडर में गैस होता ही नहीं है स्टूडेंट, सिलेंडर में तो लिक्विड होता है। सब सिलेंडर में लिक्विड होता है, जब आप प्रेशर बढ़ाते हो तो वो एक्सपांड होके लिक्विड का एक्सपांशन होके वो गैस में कन्वर्ट होता है। द नेम इज गिवन लिक्विड पेट्रोलियम गैस, लिक्विड का गैस में कन्वर्शन होता है, दैट इज द जूल थॉमसन इफेक्ट। एंड दिस इज यंग लाप्लास इक्वेशन, दैट इज इन योर आल्सो सिलेबस। At the curvatures, pressure is inversely proportional to the radius of curvature. This is very critical concept to understand for the students कि curvatures पे pressure inversely proportional होता है radius of curvature को, right? So let us check whether it works or not. Joule Young Laplace equation, ये एक ट्यूब है स्टूडेंट्स, इसका ये एंड ओपन है, ये एंड ओपन है, दोनों एंड्स पे हमने बलून लगाया है। मैं पहले इसको क्लोज करके स्टूडेंट, जब मैं इसको छोड़ूंगा तो कहां से हवा कहां जाएगी? बड़े बलून से छोटे बलून में जाएगी या छोटे बलून से बड़े बलून में जाएगी? Young Laplace equation, turn on your microphones, turn on your audio, students. Please answer. It's very interesting phenomenon. Dear students, please turn on your audio. ये इक्वेशन ऐसा है स्टूडेंट कि एट द कर्वेचर्स प्रेशर इज इनवर्सली प्रोपोर्शनल टू द रेडियस ऑफ कर्वेचर। बड़े बलून का रेडियस ज्यादा है, छोटे बलून का रेडियस कम है कर्वेचर। तो जब मैं इसको रिलीज करूंगा तो हवा बड़े बलून से छोटे बलून में जाएगी या छोटे बलून से बड़े बलून में जाएगी? प्लीज आंसर, आई विल नॉट प्रोसीड। कुछ भी आंसर चलेगा, उल्टा पुल्टा सीली आंसर चलेंगे। Am I audible? Am I audible to all of you?
Yes, sir.
प्रणाली मैडम, यू जस्ट आंसर। आपको क्या लगता है जब मैं ये दोनों को छोड़ दूंगा, यहां से रिलीज कर दूंगा, तो हवा छोटे बलून से बड़े बलून में जाएगी या बड़े बलून से छोटे बलून में आएगी? प्रणाली मैडम,
Just air can transfer both the balloons, sir, at that time.
मीन्स सेम साइज में आ जाएंगे, सेम वॉल्यूम ले लेंगे दोनों।
But the different proportion of the air, both these balloons.
सीधा एक कोई भी आंसर दो कि इधर से इधर ट्रांसफर होगी या इधर से इधर, फ्रॉम बिगर बलून टू स्मॉलर बलून और स्मॉलर बलून टू बिगर बलून।
Sir, in equal proportion, but
Okay.
first it will be start from bigger balloon to smaller balloon.
Bigger balloon to smaller balloon. Okay, so this is our general thinking. हम ऐसा ही सोचते हैं, बट साइंस ऐसा नहीं सोचता है, साइंस ऐसा नहीं करता है। Young Laplace इक्वेशन उसी के लिए आया था, जैसा हमें लगता है वैसा होता नहीं है। लेट अस चेक। छोटे से बड़े में जा रही थी, स्मॉलर टू बिगर, ये पूरा खाली हो गया है ना, इसकी हवा पूरी निकल गई। सो इसका प्रेशर ज्यादा था। यंग लाप्लास इक्वेशन वही कहता है जो आपको स्क्रीन पे दिख रहा है ना, कि एट दी कर्वेचर्स प्रेशर इज इनवर्सली प्रोपोर्शनल टू दी रेडियस ऑफ द कर्वेचर। जिसकी रेडियस कम उसमें प्रेशर ज्यादा, प्रेशर ऑलवेज फ्लो फ्रॉम हायर प्रेशर टू लोअर। ये दिखने में छोटा था लेकिन इसमें प्रेशर ज्यादा था क्योंकि इसकी रेडियस ऑफ कर्वेचर कम थी। दिस इज एक्चुअल योर एयर लाप्लास इक्वेशन इन योर syllabus in physics. So this, and let us sum up with this very interesting phenomenon, that is 2023 Nobel Prize, which is on the ottoseconds. अभी तक हमने जो सुना है सेकेंड, मिली सेकेंड, उसके बाद माइक्रो सेकेंड, नैनो सेकेंड, पिको सेकेंड, फेम्टो सेकेंड, ये है ऑटो सेकेंड। इन्होंने क्या किया स्टूडेंट, ये तीनों साइंटिस्ट ने, दिस इज फॉर स्टूडेंट्स स्पेशली। अब तक इलेक्ट्रॉन दिखता नहीं था एटम में कहाँ है, बस घूमता था, इतना फास्ट घूमता था कि कोई उसका पिक्चर नहीं ले पा रहा था अंदर। इतनी स्पीड थी उसकी, जैसे हमारे आंखों के सामने से 1000 किलोमीटर की स्पीड से ट्रेन निकल जाए तो हम उसका फोटोग्राफ नहीं ले पाते, हमारा शटर स्पीड उससे मैच नहीं होता। तो ये तीनों साइंटिस्ट ने ऐसा लेजर पल्स बनाया जो इलेक्ट्रॉन की स्पीड के साथ मैच होता था और इलेक्ट्रॉन का एग्जैक्ट पोजीशन डिटेक्ट वो कर पाए उस कैमरा से। सो दैट इज ऑटो सेकेंड 10⁻18 सेकेंड्स। सो हमारे यूनिवर्स की उम्र है स्टूडेंट्स 10⁻18 सेकेंड, और ऑटो सेकेंड का टाइम है 10⁻⁻80 सेकेंड। सो दिस इज स्मॉलेस्ट ऑफ द स्मॉलेस्ट यूनिट ऑफ द टाइम, दैट इज 10⁻⁻80 seconds. So this is very इंपॉर्टेंट इन्वेशन। अब इसके बाद पता चलेगा एग्जैक्टली एटम होता कैसा है, उसके अंदर इलेक्ट्रॉन्स मूव कैसे होते हैं, उनके कैरेक्टरिस्टिक्स क्या होते हैं। क्वांटम एज में और बवाल आने वाला है, आपके कंप्यूटर्स फास्ट हो जाएंगे, आपकी मेडिकल इमेजिंग टेक्निक्स फास्ट हो जाएंगी। So this is one of the landmark in the physics. So let us end with this. Thank you, Thierry. Thank you very much for your kind patience. This is not the end. This is the beginning of something new. Thank you.
Thank you so much, sir. You are guiding us by using and designing our own role models against high-cost apparatus and instrument for a classroom and labs for focusing and to correlate how radiation and the waveform is associated with the surrounding medium, which effects and energy takes place. Thank you once again, sir. Like the silkworm, you have built a cocoon around yourself who will save you, burst your own cocoon and come out as a beautiful butterfly, as a free soul. Now, I would like to invite Miss Dipti Ambildukar Madam, Assistant Professor, Department of Zoology, to introduce a resource person of today's seminar, Dr. Asidhara Dharvikar Madam.
Thank you, madam. It is my privilege to introduce Dr. Asidhara Dharvikar Madam, Head and Assistant Professor in the Department of Zoology at KZS Science College, Brahmani, Kalmeshwar. Dr. Dharvikar Madam is an accomplished entomologist with over 9 years of undergraduate teaching experience in classical and applied zoology, and more than a decade of expertise in training farmers and students in apiculture and sericulture. Her research primarily focuses on economically significant insect species, while her broader academic vision emphasizes environmental sustainability, student empowerment, and bridging the gap between science and community welfare. Her contribution extends beyond the classroom with over 25 research publications, 5 book chapters, and 2 patents, including innovation in silkworm disease management and sericulture technology. Madam has also registered
two UK design, one for beekeeping monitoring system and another for mulberry silkworm cocoon sorting and deflossing machine. Dr. Dharvikar's excellence has been recognized with several awards, including Avishkar Award in 2007 and Reverend S. Hislop Memorial Gold Medal for Best Paper in 2006 and multiple distinctions at national and international conferences. Madam has also contributed significantly to environmental impact assessment through her work at NIRI and other research organizations. An impressing and inspiring educator and researcher, Dr. Dharvikar Madam continues to play a pivotal role in advancing zoological science, fostering sustainable practices in apiculture and sericulture, and mentoring the next generation of scholars and practitioners. Now, I invite Dr. Asidara Dharvikar Madam to share her views.
Thank you, madam. Am I audible to you?
Yes, ma'am.
Thank you for that lovely introduction. It finds me in a very proud feeling that I have done so much work. Thank you so much. Good afternoon to all of you. I would like to extend my gratefulness to Dr. Archana Chaudhary, madam, who is the principal of Nutan Adarsh College. The chief guest of today's function, Mr. Satish Dalal, sir, Dr. Sanjeev Kondekar, sir, and I also thank the convener of the program, Ms. Madhuri Varbe, madam, Dr. Chandra Hassan Nandanwar, sir. I am very thankful to the organizing committee of the Nutan Adarsh College who have invited me here, and thank you for having me here. So can I have my presentation or I present it myself?
Yes, ma'am.
Yeah. My presentation is with someone or I should present it from here?
You should present.
Okay, fine. Okay, fine. Thank you so much. Yeah. Just let me know whether it is visible or not.
It is visible.
It is visible. Okay.
Yeah, it is visible.
Yeah. Thank you so much. The entire screen is visible, is it? Yeah. Okay.
Yes.
Yeah. So I begin my presentation. Basically, it is not a presentation. It is actually just a discussion between two groups of sciences. Rather, I would like to go back to several years when we were studying. And during our study, we always had a conversation or a debate between the physics students as well as the zoology botany students. So it was always that the win-win race went to the physics people because they said that everything that happens is because of the physics that is there in the entire universe. So we always used to say that no, it is because of the food that we eat. So we keep on discussing these particular subjects till we got to know that there's a subject, a new subject that is called as biophysics now. Right. So please interrupt if any, if I'm not audible or if there's a disturbance or whatever, especially the students, whom I would love to interact. As Dr. Ritesh Kohle, sir, interacted, what a wonderful lecture he has presented from his laboratory. I wish to congratulate him if he's there on stage. And I would like to tell him that his passionate work towards physics is definitely seen and inspiring to all the scientists around entirely in the Nagpur region, right? Thank you so much for explaining the effects that you have explained and about the autosecond also, which is a very, very new project. Thank you so much, sir. So I begin. The outline of my particular presentation today was based on the innovative science, which is an accessible tool in biophysics. So I went and dealt with the problem, which is called as any particular tool, which is very, very difficult to acquire, and we need to pay a lot of money for every aspect here, right? Because understanding zoology based on the basic rules, and these rules definitely begin from the vitamin M, which is called as money, right? So definitely understanding life through physics is the meaning of biophysics. Now, first of all, we would like to thank Dr. Kenneth Cole, who is the father of the biophysics, who has really done the pioneering work in applying physics concept to the study of living cells, where he has described about the electrical properties of the nerves and the voltage clamp technique that he has developed, understanding the nitrogen pump and the nitrogen theory, for which he received the Nobel Prize in 1947. So we should be thankful to him. Basically, the biophysics term was given by the Carl Pearson in 1892, but at that time it was not much I think, appreciated or not much worked upon. And since Dr. Kenneth Cole represented biophysics work, and therefore we got the term as biophysics, and now it is an entity, giving us both the subjects coming together, zoology and physics. I hope everyone is there and hearing me, please. If you can let me know. Yeah.
Yes, ma'am.
Yeah. Yeah. Thank you so much. Yeah. So, what is basically biophysics? If we say that we can see physics in life or physics of life, whatever is living basically depends on the physics. Like the law of physics, which we know, when the apple had fallen on someone's head, who can tell me who was the person? If you can be interactive, please. Thank you so much. Yes, if anyone can tell me.
Sir Isaac Newton.
Yes, thank you so much. Who's this? May I know your name, please?
My name is Paras Kaushik.
Okay, thank you, Paras. Thank you so much for being attentive. And this was the law that was representing a fruit that has fallen on the head of Sir Isaac Newton. And there where the rules began, I think. And basically, when we talk about the connections between the subjects like physics, chemistry, botany, zoology, uh, do you think that all the subjects are interconnected? Paris, can you answer this? Can someone answer this? Do you think they are related? Yes. Anyone can answer, please? They are connected. And if they are not connected, then there will be no life, right? This is what we study as an ecosystem. When we talk about the animals, the plants, they cannot survive if there is no atmosphere. And if there is no atmosphere, then there are no chemicals in the atmosphere. Chemical in certain ways, that the gases that we are inhaling, if there is no oxygen, if there is no atmosphere, there is no oxygen, right? So everything here is interdependent. So almost all the discoveries that must have happened in the due course of time, where science was blooming in the Western countries as well as in India, where we know that the Rig Vedas, the Yajur Vedas, and all the Vedas have all these types of sciences in between them. And therefore, they are definitely interconnected. So, what can we understand is that the biophysics will always deal with the tools of understanding living things, and living things in certain way from the tiny cells where we can complete the entire systems and the structures of the animals, right? And many discoveries that must have happened through the observations and the clever thinking of the people, where they had these available tools and we could describe what these particular techniques have been. So, aspects in zoology, if we describe the aspects, basically it's a bridge between the biological sciences and the physical sciences. So, on the basis of gravity, if we talk about, if there is no gravity, there will be nothing growing on the land of Earth, right? So, whatever study we have to begin, we have to study on the basis of the physics. So, uh, biological studies, the processes of molecular structures, the organizational levels, all depend upon the zoological structures. So, why biophysics in zoology? So, we can understand the animal physiology, the muscle, the nerve senses, and definitely the biomolecules that are we can say definitely in our studies these days, because DNA, proteins, and enzymes are particularly enhanced now, and the study of these DNA and enzymes and proteins are dealing with a lot of things going on. So, this field is particularly important in zoology because it allows us to bridge the molecular biology, physiology, and ecology with physical principles. For example, we can apply laws of optics and spectrometry and spectrophotometry to measure the DNA or the protein concentration, the physics of magnetism to visualize the brain structures through the MRIs, or the principle of mechanics to analyze the animal movements and the muscle contraction. In short, the biophysics offers an undefining framework that connects biology and physics, chemistry, and mathematics, helping us to understand animals at multiple level of organizations. One of the most influential contribution of biophysics has been to be the microscopy. But before that, we can have a broad category of biophysics tools. Now, the structural tools that are microscopes, we are very, very fortunate to be born in the era where we have microscopes and we can definitely see the structures of the cell organelles. The cell organelles like the nucleus, the plasma membrane, the cytoplasm, the nucleolus in general, the powerhouse of the cell, the energy giving cells. So everything are visible with the microscopes. Other techniques or tools that we can talk about is the spectroscopy, image tooling, where we can see the CT scans, the MRIs, the PET tests, which are prevalent these days. As we know, the cancer cells are growing in a lot of human beings, and we have cancers in everyday life these days. Every two houses, we see someone was suffering from cancer or someone is suffering from diabetes. So all these diseases can be image tools can help us doing it. The molecular tools like the electrophoresis, the calorimetry, and the biomechanical tools like the force sensors and the motion analyzers. So all these are the biophysics tools that can be used and opted in zoology. The biophysics in zoology uses the principles of physics to understand the physical, mechanical aspects of animal life from the molecular level to entire physiological systems. So it can provide a quantitative framework for analyzing, adapting the behavior and the physiological processes. The basic example that we can see, the everyday life example, as we get lots of milk at our places, which is coming from a lot of brands called Amul, Dinshas, and whatnot. Now we have Mother Dairy as well. So from where are we getting this? We are getting from a livestock or the buffaloes and the cows that we have in our dairy farms. So establishing all these techniques of multi-ovulation embryo transfer technologies have given us better cows and better buffaloes, where we can definitely get a good quality of superior quality of milk that we are attaining. So this is the major fact that we are now knowing that biophysics is giving us. Through the entire process of biophysics, we are getting this application. The second application is the in vitro fertilization techniques that we have known through the biophysics. This is a human application where the infertility uh, uh, can be restricted and the joy of giving birth to a child can be attained. So basically, these are very important techniques, or we can say many students must have been studying all these techniques right now in their semester 4, semester 5, right? So perhaps the most accessible development that I say is about the microscopic work that is being done through the biophysics, which can generate a wonderful image of cellular and molecular structures with dimensions from the microns to the nanometers. I would like to involve the DNA structure that was given by Watson and Crick, and the model was described in 1953, where they got a Nobel Prize as well. So without these, it was very, very difficult to understand how DNA is working. And on that aspect, we can now possibly see all the individual molecules, the cellular structures using the atomic force, the electron, confocal fluorescence microscopy as well. So imaging and microscopy wins the race, in the biophysics. So in the microscopy tools, we have a lot of tools here. The light microscope, I think every college of the Nagpur and other areas in Vidarbha and Maharashtra consists of a light microscope in their laboratory. And thanks to the government and the funds that we have a good, a sponsorship from the various laboratories as DBT and the science project giving UGC, who have given a lot of good microscopes in every laboratory as such. And technically, we can understand the tissue studies and histological slides that we have. Becomes an easier way out to understand the physiology and histology of the animals. And I would like to describe my work here, where I immensely work in looking for lentic and lotic water bodies, where I see the eutrophication value of the water bodies. And these water bodies showing the ecosystem of phytoplanktons and zooplanktons and the fishes as well, where I describe the Shannon-Winner index and Palmer pollution index, where we can describe the health of the lake. Now, if we talk about lake and pond, why they are important? They are important because they are an integral part of our irrigation departments. If these irrigation departments don't give us waters, the corporation would not give us the water. Now, this water has to be cleaned in a proper way. And giving this water, which is filled with sewage or which is filled with such organisms, makes the health suffer in lots of houses. Like this season of July, August, and September, we have a lot of influx of sewage water, and due to which we have a lot of diseases, right? So we can introspect these waters, and we can develop the studies of seeing these waters under the microscope. And definitely we can find these cladocerans who are cleaning the water, the phytoplanktons that are giving the water, most probably the life of the water, I can say. So this gives immense pleasure for carrying out such activities in the colleges also. So you can get some water from the ponds which are in your near vicinity, and you can check it under the microscope, where you can see a lot of phytoplanktons as well as zooplanktons. The other structures that we can also see in the biodiversity world, like we have lots of insects around us, we have lots of butterflies around us. So we can keep on looking for the scales on them, as well as the fish scales and the insect wing scales, which can definitely produce a lot of amount of papers and inspiration and inclination towards the zoological studies. And we can also have images of them and carry out a lot of research work. Other aspect of biophysics which has really acquired a lot of interest is the transmission electron microscopy and the scanning electron microscopy, where we can see the ultrastructure of the organelles. So immensely zoological researchers have put themselves inclined towards all these subjects. So this becomes the winner of the biophysics, I should say, microscopy. Next come the spectroscopy tools, where we can find the protein nucleic acid concentrations, whereas the principle of the UV vis spectroscopy, the molecules absorb light in the ultraviolet radiations 200 to 400 nanometers, where the visible 400 to 700 nanometer region of the spectrum. Now, the protein concentrations, as we see, is around 280 nanometers, due to which the aromatic amino acids like tryptophan, the tyrosine, the phenylalanine, all these amino acids can be absorbed in this particular spectroscope. And definitely we can measure their absorbance with the help of estimated protein concentrations. So here we use the Beer-Lambert law, which is the most important law, and helping us as zoologists, telling us the nucleic acid concentrations, basically studying the DNA and RNA, which is absorbed at the maximum 260 nanometers because of their nucleotide present in them. Nucleotide bases, the 260 and 280 ratio is also used for the assay purity, whether the proteins that we have got from some particular animal are contaminated or not. So that can also be visualized and can be tested through this spectroscopy tools. Next is the fluorescence spectrophotometry and the spectroscopy, where the enzyme activity and the cell imaging activity can be seen. Here, an emulsions are made which are tagged with the GFP protein, the green fluorescence protein. And when this enzyme acts with the particular fluorescence, giving us a particular substrate. And these substrate are fluorescent dyes. And these dyes go and track the molecules and the organelles which are living cells. So whenever they are tagged with this GFP, they glow under this fluorescence microscope, telling us the presence of the viruses and the bacteria in that particular area. For instance, I can tell you that I was working on the nucleopolyhedrovirus, where the silkworms were affected with the grassery disease, and the farmers during the crop of silkworms, when the silk cocoons had to be made, they were diseased. So the farmers couldn't get good quality of the cocoons. So my basic study was on green fluorescence protein, where I tagged these viruses, the nucleopolyhedrovirus with this GFP, and around 7 generations together, I could see the green fluorescence protein coming from their mother as well as the father. So I took up the study of venereal transmission as well as transovarial transmission, where I proved that both the parents are involved in carrying this virus, as it is a nucleopolyhedrovirus, which was not similar with the cytoplasmic polyhedrovirus. So in the cytoplasm, the green fluorescence protein was not tagged. So In the nucleus, when it was tagged, it was passed on to the generations together. So believing that the viruses are inside these cocoons and therefore the adults also carry the viruses, and in the certain conditions of atmosphere, they definitely get this infection of the grasserie. And this is a major fact of biophysics, I should say, that it has helped to prove a lot of diseases, disease carrying parasites, and so many things that we now know about. Infrared spectroscopy also shows the biomolecule vibrations. Now, these vibrations are caused because of the bonding vibrations. And applications for the biology is that it helps the identifying the functional groups and study structural changes in the protein, lipids, and carbohydrates. If any particular protein is being made and this protein is affected, or the person is affected with some disease, then therefore the particular protein is also affected by this. So this can be shown by the bond vibrations also. The next technique that we study in our MSc as well as in our BSc classes, where we see the electrophoretic techniques, where the gel electrophoresis, where we can determine the DNA protein separations according to their molecular level. Then we go in for the SDS page, that is sodium dodacyl sulfate polyacrylamide gel electrophoresis. This is for the protein profiling, whether the protein is exactly with the control that we give. So this application is definitely used for the genetics and the biodiversity studies, as we are getting a lot of viruses and the bacteria that are mutating. So now these mutating bacteria as well as viruses can be gel electrophorecized as well as SDS phorecized, and we can get a protein profile with it. And this protein profile would definitely land up with uh, some type of imaging, as well as the we can say like the protein profiling, the phylogenetic analysis. The phylogenetic analysis will determine the databases of earlier diseases also. So we can find out whether the virus is a variant one or it is a mutant one. Okay, so this is a very good technique that we can find out in our zoological techniques. Then we have the imaging. So imaging is very, very important these days. For example, I talked about the PET test. Even your principal madam talked about the PET test, which is now a verified test for the cancers. Now, this PET test is positron emission tomography. And this tomography explains about the metabolic activity in the living organism. So whether the cancer cells are spreading from one part to the other can be determined by this particular PET test. And other tests which are very, very similar, like the CT test and the MRI, which are now quite old tests, and we are going through all these tests whenever we are getting injured or whenever we are getting some type of disease. We can definitely go and do our CT scans and MRIs. For example, anyone, if they have a head injury, definitely they should go in for a magnetic resonance image, that is MRI, to describe the brain mapping. So this can be also done with the other animals. The disease models also can be run through all these things. So this is also a very good tool for the particular from the biophysics for the zoology people. The other biomechanic tool, that is the force transducers. These are the study muscle contraction techniques by the animals, and muscle contraction in sense of movement of their body parts, like their hands or legs. So whenever we talk about insects, the insects having two pairs of wings and three pairs of legs. So the movement of their body describes. And in the laws of biophysics, if you can see, many of the vehicles that today we are driving, whether it is the aerodynamic model of the scooties that we are using, the two-wheelers we are using, and as well as the aeroplanes and the helicopters that are being used by the defense and other countries. They are inculcated from the insects, and these insects are being studied in depth with these particular techniques, and therefore now we have new models of all these helicopters and aeroplanes, and definitely they are helping us. Even zoological aspect is being taken up by the biophysics people, and they are learning these techniques, how these animals are working and how the locomotion is being done in the insects, how these birds are moving from one area to the other area, the motion capture systems. So with this we can conclude that these particular biomechanic tools are helping in our day-to-day lives. So this is one of the example that I would like to quote, the biomechanic tools also. The others that we are already known of, the bioinformatic tool. Now this bioinformatic tool is definitely with everyone. Everyone who is having a smartphone can go in for the sites of the bioinformatics, where you can find the computational tools. And now these computational tools can give us the DNA structures, the protein structures, and the enzyme structures. Now, what is the purpose of visualizing them? Visualizing these will definitely let us know how these molecules are working, whether they are interacting with a particular organ, they are uh, they are influxing their particular solutions towards some of the organ or not, if they are being interrupted by some particular other organs, enzymes they can be. So what is the central dogma of life? The DNA, the replication of the DNA, the transcription of the DNA and getting an RNA, and then the protein. Now when we are talking about the central dogma, we can also visualize or understand how the insecticides are interacting with the insect proteins. Now, why I have taken this particular insect proteins? Because in today's day-to-day life, whatever we are eating, we are consuming, consists of a lot of pesticides and insecticides. Any vegetable you take contains some nanoparticles or some microparticles of these particular insecticides, and definitely they are harming our body. How are we going to know about this? We definitely have an answer to this, because the cancers are coming up. Basically they are interrupting in some or the other systems of our body. So stimulating enzyme activity will be helpful, and we can do this with the help of the bioinformatics. We can study the disease resistance as well as the drug present there. If we can give the drug, we can prepare a drug, and we can use these drugs and see whether they are really giving us the results. And I have also quoted for the parasites, because the parasites are also endoparasites and ectoparasites. Both are showing some type of disease resistance as well. So if we talk about the pests, the mosquitoes, the house flies, now you can believe that the NMCs are spraying a lot of insecticides, because we have a lot of amount of mosquitoes these days, because of the water, because of the rains. So we are dealing with these problems day to day. So we should definitely have an inclination towards the study. The phylogenetic analysis can also give us the relationship between the evolutionary part of the animals that we have. It is said in the evolutionary manner that we came from the New World monkeys, and New World monkeys got converted into humans, right? So do we have a phylogeny? Yes. So this phylogeny is being proved with the help of the bioinformatics. And now the relationship has been made, it has been justified, and definitely we have the collection of the data. And this data, evolutionary data, is now telling us or giving us a family tree, that definitely we are coming from and we are sharing most of the type of DNA that the monkeys are having, right? And we are also sharing our DNA with so many other animals, right? So this has been proved with the help of the phylogenetic analysis by the bioinformatics. Now, how this data can be collected? You must be saying that, ma'am, this is a very, very expensive work. But definitely, if you can do once, if you can take DNA from an organism, you have to collect the data, you have to collect the DNA, the protein sequences, and the database. You have to align them with the gene bank. And definitely, we have so many other softwares who can tell us about the tree construction, the mega software, that is the molecular evolutionary genetic analysis, the beast one. So it is like it is likely that all the phylogenetic estimations can be diverged, and we can definitely find out the common ancestors as well. So interpreting these common ancestors and the evolutionary chart will again bring a big boom in the zoological world, right? So these are some of the phylogenetic analysis work. So I went through a paper which I came across where the comparison of the genomic multi-tools for the scientific discovery has been made. And here in this Zoomania project, we can see most of the eutherian families, they are representing in almost one particular way. 83% of the DNA is being acquired and is being similarized, and it is similar to human DNA. So you can also go through this particular paper and you can visualize. Yeah. Other applications that we can understand is the different types of insects and birds, how the mammals evolved through the bioinformatics, the conservation of the biology, identifying them genetically, whether they are now, if we talk about the endangered species, we can acquire their stem cells, store them. The frozen seeds are there. We have a lot of applications these days. We can keep such type of stem cells in different laboratories, and we can assimilate these information. Rather than they being endangered, we can bring them back, like the projects that are happening for the green turtles, the projects that are happening for the leopards. It can also be done in our area as well. So, and confirming of the classification of the new species can also be developed with the help of this bioinformatics tool, right? Disease tracking can also be done because lots of disease has been the COVID-19 as we have gone through. So why and how these particular viruses have come? Now, these viruses have come from the animals, it seems. Now, these animals are carrying them since so many years. So we can track these diseases, and the study of these diseases can be done easily with the bioinformatic tool, right? So the protein structure determination can also be done. I have an example here. We can trace the butterfly evolution by comparing their genes with the other genes. We can also have the diversity of the honeybees. Like we have only 5 particular species of honeybees in India. Why not others? So what is the diversity of the regions that we can always talk about here? And also,
इतकं मोठं उदाहरण म्हणजे आजचा विक्रम सिद्ध करतो की विद्यार्थ्यांची कल्पना, त्यांची मेहनत आणि त्यांच्या चिकाटीच्या जोरावर आपण जगाला दाखवू शकतो की ज्ञानाची खरी शक्ती कशामध्ये आहे. आणि म्हणूनच त्या विश्व विक्रमाच्या प्रवासामध्ये आपण करूया इतकं देखील मार्गदर्शन करायला.
Yeah, interrupted. So we also have one more technique that is called as wildlife forensic techniques, which can be used in zoology. So I think the students might be knowing about the PCR techniques. So where we can amplify the DNA coming from the hair, the skin, and the bones, as well as the blood. So by the DNA fingerprinting, we can come across the poacher cases, the poacher cases who have taken the several parts of the animals, the skin, the tusk, the horns, and the skin of the particular animals. The databases can be acquired, and the confiscation can be done, and the illegal trade monitoring can be detected with these techniques. So the court evidences has to be proved with the help of the DNA fingerprinting. So this can also be done, and the scientific proofs for the wildlife crime cases can also be evident with the help of this forensic sciences. So this can also be included as a biophysics technique that is there. Emerging tools, we have a lot of emerging tools coming up. The NEP has come up. The NEP has a lot of interdisciplinary project work as well as interdisciplinary subjects. So in the scenario, we can also talk about the electron microscopy, where we can visualize the proteins in the animals, and the 3D images can be seen. The control images can be seen and compared with the diseased ones, which can give you an insightful and remarkable studies in developing and the vaccines as well, right? So for the disease mechanisms, we can use this cryo-electron microscopy technique. One of the studies also show the hemoglobin in the animal, the structures of the hemoglobin in other animals, and their insight in the oxygen transportation. The other emerging tool is the optogenetics. Now, optogenetic deals with the control in the neurons with the help of light. Now, here the genetics and the optics are combined, and this is a very, very flourishing topic. I would like to tell you, students, definitely you can have a lot of AI information about this on your Google, and as well as the brain behavior, the circuits of the brains, the learning and the memory techniques, the immunology techniques can be easily demonstrated with the help of this optogenetics. So dive your interest in this, students, so that you can have an inculcation towards the biophysics. The other nanotechnology in the animals, where we can see the engineered nanoparticles. If we send these particles as medicines, as well as healing drugs, like the delivering of the anti-parasitic drugs that are used for the infected livestock. Now, livestock have a lot of parasites inside them. So definitely they are being injected, and these injections are given with the help of proper dosages. These are disease diagnostic way they have given them, and this is another new studies that have been found out. And I would like to tell you that there are a lot of challenges and limitations to this study. They are immensely costly. The instruments are costly, but definitely during the development of sciences and incubation centers, we can have these particular technologies in our departments as well. And now we have affordable perspective also, where we can have a biophysics tool, which is AI generated, machine learning. We can have applications in the conservation biology as well. So we have to deal with these problems, and definitely we have to survive with the challenges and the limitations. The summary of my biophysics work, or biophysics as such, is that it provides a definitely very good tool to the entire study of organelles, the study and applications of the genetics, the physiology, the ecology aspect, the environment conservation aspects, and it will be an compulsory tool. I tell you, it will be a compulsory tool for the future zoologists and the researchers. So the teachers, the faculty members who have joined recently, definitely dive in these subjects. So the discussion part, I would like to ask you, students, what tool can be used by you? Okay, so this is a question I leave with you, and definitely you are going to get your answers till you pass your UG and your masters. Okay, thank you so much. So exploring biophysics tool has a deeper understanding of zoology, and every one of us is a powerhouse. We all have a mitochondria in us. So this mitochondria is continuously giving us energy to sustain in this world. So I leave you with this idea of biophysics coming into your life, and I hope the progress in your life definitely depends on the biophysics. I thank you, all of you, for having me here, and thank you, organizers. I hope the study or the biophysics PPT is helpful for all of you, and thank you so much.
Thank you, madam, for guiding us your stimulating and energizing words that how can use instruments and tools in sericultures and apicultures. Now we have moved towards the questioning of this today's seminar session. If any participants has any question, then please raise your hand virtually, or you can type your message in the chat box. Unmuted, all the participants unmuted and ask the questions. There is no questions, if anyone.
It means the information was not good enough. If they would have had a question, it would make us believe that you have all gone through the presentation. Yes?
Yes.
The presentation was so good that all the questions are answered.
Oh.
The flower doesn't dream or beam, it blossoms and becomes. I would like to invite a chief guest of today's seminar session, respected Mr. Satish Dalal sir, Head and Associate Professor, Department of Zoology of Nitin Adarsh College, Umred. Dalal sir.
Yes.
व्यक्तिगत सोल्युशन हा क्षण फक्त त्यांच्या आयुष्यातील नाही तर संपूर्ण समाजाच्या शाळेच्या आणि आपल्या भारतासाठी सुद्धा आणि वर्गचित्रासाठी सुद्धा अभिमानाचा क्षण आहे. विद्यार्थ्यांना लहानचं स्वप्न मार्गदर्शनाचं आणि पालकांच्या पाठिंब्याने आज
Dalal sir,
Hello.
speaking.
Yes. Madam, मी दिसतोय?
Yes,
Yes.
sir.
Yes, sir.
Huh?
Yeah.
Can you see me?
Yeah, sir. Yeah.
Huh? Huh.
Yeah, yes,
Thank
sir.
you. Thank you. So, respected principal of this college, Dr. Archana Chaudhary, and to today's research person, Dr. Ritesh Kode, Head and Assistant Professor of Department of Physics, Sant Gadge Maharaj Mahavidyalaya, Hingna, and another dignitary resource person, Dr. Ashidara Dharvikar Madam, Assistant Professor and Head, KJS Science College, Brahmani, Karmeshwar, and all the organizing member of this seminar. So, first of all, I want to congratulate all the members and organizing committee who had made successful this seminar. In the today's seminar, we have seen
the topic in physics as well as in the biological field. First of all, I would like to congratulate Dr. Ritesh Kole sir, who has given excellent demonstrative physics talk. Sir, this physics and biology, they are very much interconnected subject. The physics here, one subject it was living and another subject was non-living. But in the today's seminar, we have made the proper connection. Sir, you had given excellent talk of physics, and this physics and zoology or biology, these are very much related subject. Here, this physics, it has solved so many problems in the biological science. I remember that in the year 1954, there was a problem regarding the genetic code, the triplet code in the 1954. It was solved by the well-known physicist Gamma, who had given first time in the 1954 the genetic code is triplet code, and since then, from 1954 today, the genetic code it has remained unchanged. In this refresher course, here the physics, the refractive index of glass which we use in the micro technique, it is very important. Now the role of the light you have told here. So sir, the light it is an important factor in the development of the biological science. Being the professor in the ichthyology in the fisheries, without light, the fishes they do not lay the egg. Here, although the fishes, particularly in the monsoon period, the female it is loaded with the egg inside her body. But unless and until there is no lightning in the sky and thunderstorm, the fishes they will not lay the egg. So this is the very important role of the light in the biological science, in the breeding of the fishes also. So here the physics it plays a very important role in the microbiology. Various instruments we are using in our department such as oven, fridge, autoclave, etc. All these are running by the law of the physics. Here the various type of the rays which such as the alpha, beta and gamma, they play a very important role in the mutation in the life sciences, in the biological sciences. So this light plays a very important role in the life of the plant as well as the animal. So, sir, you have excellently demonstrated the waves, the how the waves travel. You have shown a small instrument and demonstrate. So here you have done the excellent work. Then with regarding to the energy, kinetic energy and the potential energy, how they work, that you have also told. Now, beside that, the ideal gas equation which we had learned in the 12th class. So this ideal gas equation PV is equal to nRT. So here all these important laws in the physics, all these are very fruitful, they are useful in the biological science, and for you have demonstrated very well. So for this purpose, I congratulate you, sir. Then another resource person, Dr. Asidara Dharvikar madam. This madam, you have told that all the subjects they are in the science, all they are the interconnected. No single subject is away. All the subjects they are interconnected. And here the relationship between the biology or biological science and physics. You have explained very well that various spectroscopy, MRI, and various biological tools, you have also throw the light on this instrument. Then with regarding to the DNA, the structure of the DNA, Watson and Crick model, that too you have told. And regarding this health of the water body, the pond water body here, and the role of the plankton, phytoplankton, zooplankton, here all you have explained, and also you have explained the little about the biodiversity. So here, beside that, in the sericulture, you have told that the pest, parasite, and you have also focused on the various genetic phenomena. So for this purpose, for this you are on this talk on this various subject, madam, I would like to congratulate and thank you, madam. So beside that, all the members of the college regarding this organizing this seminar, this the madam uh, from my department, Miss Madhuri Varbe madam, the Chandrahasa Nandanwad, the from Department of Physics, they have played very well the role in the running this seminar. I would like to congratulate these two personalities also, and other members whose continuous effort by successful carrying this seminar, that is Shailendra Khambalkar, Dr. Shailendra Khambalkar, the Pranali Hinge, also from the Department of Physics, the Nilesh Chaple, all these members belonging to this Department of Physics. Here, I would like to congratulate this person. And finally, the Dipti madam, Dipti Amilduke madam, assistant professor of my department. Here, I also congratulate her for successful conduction of this seminar. Thank you. With this few word, I would like to stop here. Thank you. Thank you, everybody.
Yes, Hinge madam.
Thank you, sir. To express a brief summary of today's event with your divine voice. Finally, I would like to invite Dr. Shailendra Khambadkar, sir, Head and Assistant Professor, Department of Physics, to express a vote of thanks.
Hello. I am audible.
Yes, sir. You can start now.
Good afternoon to everyone present here. On the behalf of organizing committee, it is indeed my privilege to extend a heartfelt vote of thanks at the conclusion of this enriching one-day national seminar on innovating science, accessible tools in biophysics. First and foremost, I express my deep gratitude to our chairperson, Dr. Archana Chaudhary Madam, Principal, Nutan Adarsh College, Umred, for gracing this occasion with their valuable presence and inspiring us with the word of wisdom. I express my sincere thanks to our esteemed resource person, Dr. Ritesh Kohli sir, and Dr. Ashidara Dharvikar madam, whose provoking address has set the tone for today's discussion and added the value to the seminar. They shared their expertise and insight with great clarity and depth. Both of your contributions have made this seminar truly meaningful. The knowledge share will motivate our participants towards the innovative scientific approach. My special thanks to our chief guest, Mr. Satish Dalal sir. Your inspiring words, encouragement have truly set the tone for today's seminar. We deeply honored by your support and guidance. I extend my warm thanks to the IQAC coordinator, Dr. Sanjeev Kondekar sir, whose vision, planning, and constant guidance have been backbone of this seminar. I would also like to thank our convener, Ms. Madhuri Varve madam, and organizing secretary, Dr. Chandrahasya Nandanwar sir, for their tireless effort and constant encouragement and support in organizing this event successfully. I would also like to acknowledge the effort of our organizing team members, Dr. Khamadkar sir, Pranali Hinge madam, Nilesh Chaple sir, Dipti Ambiluke madam, whose dedication behind the screen ensure the smooth conduct of the seminar. I would like to thank our anchor, Ms. Pranali Hinge madam, who has made today's seminar with a grace, confidence, and perfect coordination. Last but not the least, I extend my heartfelt gratitude to all the participants, students, and delegates for their enthusiastic involvement. Your presence and active participation have made this program vibrant and fruitful. With this word, I once again thank each one of you and the Zoom platform for making this one-day seminar a grand successful. Thank you.
Thank you so much, sir. At the end, I will give you inform to all the participants about the feedback form. It will send it on your WhatsApp group. So all of you fill that form to create your certificate of this today's seminar session. Once again, thank you so much all of you to make this event memorable and such a like that we will take this event, such a type of event in future with your guidance and support. Thank you once again, and with the permission of host, we will end this meeting of this session at that time. We can leave it now.
आमचं सापडलंच नाही आमचं 3 लोकांचं ऑनलाईन